we will be covering a large portion of Chapter 6. So to make this content a little bit more manageable, we're going to break it up into 3 different videos. In Part A today, we're going to cover slides 1 through 12. Part B, we will cover slides 13 through 24, and then slides 25 through 30 will be covered in lecture video C. Let's begin by looking at nutrition. We're going to talk about microbial growth, and it's going to rely on the nutrients that are available. The rate at which bacteria divide is incredibly fast relative to other organisms. E. coli is one of our fastest dividing bacterial species. It has the ability to undergo binary fission about every 20 minutes when we have ample nutrients that are available and its environmental conditions are favorable. That's incredibly fast. Many of you who are growing bacteria in lab have seen this, where you've introduced a plate or a tube that has broth in it, and you've put bacteria on it, but you can't see any of those cells. You incubate it overnight, sometimes 48 hours, and then you pull out that media and you can see that bacteria. Well, a single E. coli cell at this rate of binary fission every 20 minutes could end up being roughly 100 trillion cells within a 24-hour period. So this tends to be one of our fastest replicating species. There are certainly a range. So there are some species of bacteria that take longer than this to undergo binary fission, but in general, bacteria will grow pretty quickly as long as they have ample nutrients. So let's talk about some of those nutrients that they are going to require, not just when we grow them up in lab, but when they're growing out in the environment, in the wild, so to speak. So we can talk about what are called essential nutrients to our microbes. These nutrients are those that the microbes cannot synthesize themselves. They don't have the correct machinery to actually assemble these particular nutrients. Because they can't make them, they require these nutrients come from their environment. And all of our species are going to need these essential nutrients. So some examples might be particular amino acids to form our proteins. So a lot of those building blocks of the macromolecules that we talked about back in, chapter 4, are going to be considered essential nutrients because they are needed to make those macromolecules. There are some cations that are going to be necessary to allow enzymes to catalyze their reactions. Without those cations, which are considered to be the essential nutrients, the enzymes won't work, and we won't have those critical chemical reactions that our microbes need. So essential nutrients can't be made by the microbes, but all of our microbes are going to need them. So when we try to grow when we try to grow bacteria in a lab, all of them are going to have these components in the media that we supply. So not all species need what are called growth factors. Some of our species of bacteria are a little picky about the nutrients that they need. So growth factors are additional nutrients that have to be supplied to certain microbes. If they're not supplied, either the microbes won't grow at all, or sometimes they just won't grow as well. So when we have media that has these specific growth factors needed, where we're targeting and trying to grow up a particular species, we refer to that media as being chemically defined. So when we are growing microbes in a lab or when they are living out in the environment, they are going to need a source of nitrogen. We are also going to focus on their carbon source that they need. We're going to focus more heavily on carbon than on nitrogen. So all organisms on Earth are carbon-based. You are, so am I, your, I don't know, pet dog, if they're in the room with you right now. They are carbon-based as well. So microbes are too. So we are all going to need a source of carbon. When we have microbes that have the ability to assemble their own carbohydrate molecules, these big carbon molecules, from simplistic carbon dioxide, we refer to those organisms as autotroph. So try, especially as we continue throughout the course of this lecture, try to break down some of these words. So the auto, the first part of this word, means self, and then troph, that second part, means feeding. So autotrophs are those organisms, those microbes that use carbon dioxide gas and take that carbon from that carbon dioxide gas and use enzymes to build a much larger carbohydrate molecule. They may build glucose, for instance. So then the organisms that take those larger carbohydrates, glucose for instance, and break them down, release that carbon dioxide. So in this way, we see a cycling of carbon. The organisms that use the larger assembled carbohydrates are called heterotrophs, other feeders. That prefix hetero means other. So let's look at a diagram that kind of represents this carbon cycle and shows us how we can change the kind of form of carbon that we're dealing with depending on the type of microbe that we have. Okay, so this is from your textbook, and it's a little more complicated than it needs to be, so we're going to kind of pick out and point the most important things out to you, okay? So here is where we see our glucose, and they've shown you behind it that it is a 6-carbon molecule. So the chemical formula for glucose is C6H12O6. I'm not concerned that you know that at this point, but that's the molecule that they're referring to. It has 6 individual carbon atoms that are part of it. Well, our heterotrophs, through a series of chemical reactions, are going to utilize these 6 carbons and break them down in a series of metabolic processes that we'll detail later in this unit. What the important thing here is to follow is this carbon dioxide that is produced by that. So we said that heterotrophs use carbohydrates, glucose is going to be our example, and they release carbon dioxide. Well, this is that released carbon dioxide. Oh, who can use that carbon dioxide? None other than our autotrophs. So if we follow this over here, our autotrophs are going to take this molecule of carbon in our carbon dioxide plus several others, and they're going to assemble it back into that big 6-carbon glucose. So back and forth, we cycle through this carbon, converting it from one form to another and back again. There are different microbes that do each of these processes. So depending on how they utilize their carbon, we can either categorize a microbe as an autotroph or a heterotroph. All right, so carbon is one of our most important nutrients. Got to have a source for it. In addition to carbon, all of our microbes are going to need a source of energy. This, again, it's all cells, so not just microbes, your cells too, but all living things have to have a constant input of energy. They need that energy to do work within their cells. Without the energy, work cannot happen, and the cell simply will not survive. It will die. So there are 2 ways in which microbes can obtain their energy, and we give them names based on the source of that energy. So the first category that we're going to talk about are our phototrophs. So those microbes that undergo phototrophy are able to capture the energy from a light source. That light energy is absorbed and used within the cell. If light isn't the source of energy, a chemical is going to be the source of energy. So if we have chemical electron donors that are being oxidized as a way to get our energy, we say that that microbe is a chemotroph. Chemo meaning chemical. Okay, so again, either photo feeding or chemical feeding. Within the category of chemotrophy, we can either be looking at the use of inorganic chemicals or organic chemicals. We use terminology to denote that difference. So if we have an organism that is a chemotroph and they use inorganic molecules as the source of their electrons, we're going to say that they are lithotrophs. So for those of you in lab, you may use special media designed to allow these lithotrophs to grow. This is how they're going to get their energy source. So we may need to provide them with things like hydrogen sulfide, ammonium, nitrate, or ferrous iron. Sometimes we can use the growth of our microbes in lab in this type of media to help us identify or maybe narrow down the possibilities if we have an unknown specimen. So only certain specimens can obtain their energy this way. Most of the organisms that you're going to be dealing with in lab are considered organotrophs because they are going to be using organic molecules to donate their electrons. The primary example of this is sugars. So we talked about one of our sugars when we mentioned carbon sources. We said that glucose, a monosaccharide, is a really common source of carbon. Well, many of our microbes are going to use it as a source of energy as well. So when we start thinking about where we're going to find a given species of microbes, their carbon source and how they obtain their energy is going to be important because those are going to be necessary things that the microbes need. So let's think for a second about the environment that our body would provide. We are carbon-based life forms. We are going to have glucose as an ample source of both carbon and energy in our bodies because we need that molecule for our own processes. Well, you know what? Bacteria who use those same components for carbon and energy are going to be the ones that are our pathogens because that's the environment that our bodies provide. So the pathogens that we see being problematic to humans are going to be heterotrophic organotrophs. They are going to use sugars, including glucose, as both their carbon source and their energy source. We will look at the metabolic pathways that detail these processes in later chapters. All right, so before I start on section 6.2, I'll just make a quick note. I'm only covering part of section 6.2. There's additional information on what is called spread plate and another section on serial dilutions. Both of those have some mathematical equations and calculations that are associated with those. I cannot remove that content from the inquisitive. So when you are doing inquisitive assignments and you see content from 6.2 come up that I didn't cover, especially those math problems, Lower your confidence level and guess. It's okay for you to be wrong. I will not ask you to perform those calculations on quizzes or on your exam for this material, okay? So I want to focus on growing bacteria in a lab, and then we're going to kind of compare how bacteria grow in a lab versus how they grow out in the environment, okay? So for some of you, you may have had the opportunity to actually culture bacteria. So the term culture is a verb. It can mean to grow up or to allow your microbes to have the correct conditions that they can undergo binary fission really, really rapidly. We can also refer to a culture, which is a media of some form that has bacteria growing in it. Okay, so that's what that term means. So we use media that has all of the nutrients that bacteria need, whether they are essential nutrients for sure, but if we have any growth factors that need to be added, we have to know that so that we can provide that particular media to our microbes. So there are a couple of different types of culture media that are used to grow microbes in a lab. The one at the very top here, this one up here, is a liquid form. We call that a broth. So if your nutrient media is in a completely liquid form, we'll call it a broth, and it is almost always going to be in a tube or a flask. So we will typically have a lid that sits on top. It is rarely going to be sealed shut. It's usually just enough to prevent contamination from the air settling into our liquid media, because if you have contamination, it's going to grow too. Any microbes that are introduced into that broth will grow. We have two forms of solid media. We have a tube over here that is called a slant. So the slant is made beginning with a liquid broth. To that liquid broth, we will add a powdered substance called agar. Do you remember where agar comes from? Agar is a derivative of seaweed, also called algae. So this is a solidifying agent. When we are making our slant tubes, or sometimes just referred to as a slant, we heat up a liquid broth, a nutrient broth. We add that powdered agarose and we let it dissolve. It's a lot like making Jell-O. Then we will pour that media into a tube, and it is cooled at a tilted angle on a slant, which is why it's given that name. A slant tube gives us a nice big surface to grow our media on, but it also gives us a deep amount of surface. So if we ever want to inoculate our tube or introduce bacteria for the purpose of growing, we can inoculate that tube by stabbing all the way down and then back up. It gives us a nice thick area. We'll talk about why that might be important later on in this chapter. We've mentioned Petri dishes before. We use several different names for these guys. Sometimes we'll call them a Petri dish or a Petri plate, or sometimes they'll just be referred to as a plate. These guys are made with the same basic principle. We have a liquid nutrient broth. We add our powdered agar. We let it cool and solidify. The lid sits loosely on top of this. Again, it's meant to prevent contamination. So it's loose and it rotates around. All right, so in lab, we like to maintain what is called a pure culture. So when we are working with bacteria in lab, we want all cells of a single species in our media, whatever it is that we're growing. Typically, in lab, we're going to be running biochemical tests, or we're going to be staining our organisms. We want to look at that species as a whole and see how it behaves. By having a single genetically identical species or a single genetically homogeneous, all the same species, it allows us to characterize that species' morphology and their metabolic pathways. So we want to test and work with and maintain our pure cultures. We want to avoid contamination. However, in the real world, out in the environment, our microbes do not live in isolation. We never have a pure culture. We have mixed species living together. So there are times where we might want to isolate one species and test it from all of the others that it might be in contact with. So we have special procedures that allow us to separate a pure culture, one species, from a mixed culture. Now, this is done in a couple of different ways. And if you actually perform one of these in lab, each lab professor has a little subtle twist on how they like to teach what is called an isolation streak. Sometimes it's also called a quadrant streak because it's not uncommon to divide your Petri plate into 4 different sections. And how that division is done and can be done is kind of a unique thing from lab to lab, but the idea is going to be the same. So with a isolation streak, our goal is to take a mixed culture. It may be 2 or it may be many different species, and we want to separate out the cells. We're going to do that using the broad surface of a plate. So I'd like to begin by watching an animation of a streak for isolation, or also called a quadrant streak, and then we'll talk through it. Some of you may have done this before. I've chosen this one because it's my favorite in terms of what I teach my students. But like I said, your lab instructors, if you're taking lab, may have a subtle twist on this.
Flame your loop. Using aseptic technique, remove a small bacteria sample from the tube. Position the Petri plate so quadrant 1 is beside your non-dominant hand. Remove the lid slightly. Starting at the outer edge of quadrant 1, streak the loop into the quadrant. Flame your loop and turn your plate a quarter turn. Cool the loop before touching the plate. Place your loop into quadrant 1 and drag it into quadrant 2, zigzagging between the quadrants at least twice. Then continue to streak the loop through quadrant 2 to cover the area. Flame your loop and turn your plate a quarter turn. Cool the loop. Place your loop into quadrant 2 and drag it into quadrant 3, zigzagging between the quadrants at least twice. Continue to streak the loop through quadrant 3 to cover the area. Flame your loop and turn your plate a quarter turn. Cool the loop, and then place it into quadrant 3 and drag it into quadrant 4, zigzagging between the quadrants at least twice. Continue to streak the loop through quadrant 4 to cover the area, and be careful not to enter into quadrants 1 and 2. Flame your loop.
All right, so did you notice that there were lines that were drawn on the bottom of that Petri dish? Why do you think we drew them on the bottom rather than on the lid? Because the lid rotates. So one of the challenging things about learning an isolation streak, or a streak for isolation or quadrant streak, all of those are referring to this same process, is the fact that you can't actually see the bacteria themselves. As you lightly drag the inoculating loop over the surface of that agar, it leaves a little bit of a trail, but sometimes it's difficult to identify where it is that you've placed the bacteria. So an isolation streak, whether it's done drawing the way that we saw in the video, the one that's depicted in your book actually does 3 sections. I very commonly will see professors who will have students draw them in like very deliberate, equally divided quadrants. So they'll have like a cross on the bottom. But the idea is that it's a pattern of inoculating where we introduce bacteria into one part of the plate, and then we go through a series of steps to essentially spread out all of the cells. The idea is that by the time that you get to the last step, you are going to have your cells sufficiently spread out that individual cells will be deposited. After you grow up your bacteria, so this one down here has been sitting in an incubator for overnight or so, we see that there's too much bacteria here. We can't see individual circles. Those circles are called colonies. That's what we want. We see some overlap where we have taken that bacteria and further spread it out. We take the bacteria and continue to spread it out over here. What has happened is that at each of these colonies, we have had a single cell deposited. When that plate is incubated, that one cell will undergo binary fission and divide and divide and divide and divide. It'll do it so many times that overnight we will be able to have sufficient bacteria grown up that will actually be able to see the cells. Now, while a colony in theory represents one cell that was initially deposited, we have to be a little more realistic and say, yeah, you know, sometimes there are small clumps of a species that are all sticking together. So to be more accurate, we will say that a colony was formed by a colony forming unit, or a CFU, rather than just saying that it was a single cell, because we can't be sure that it was just one cell. But we do make the assumption that all of the cells that are within that colony originated and are of the same species. So it's important that you understand with our streak for isolation, we introduce bacteria into one part, and then each time she flamed that loop in our video, did you notice that it gets bright hot red? Well, that incinerates all of the bacteria that were on the loop. So when she touches it back down on the auger where there was an overlap from the previous quadrant, that allows her to pick up just a little bit of bacteria and spread it out. So we introduce a lot of bacteria in the first section of our plate, and we go through a series of steps to separate out and reduce the amount of bacteria so that we can get to individual colonies being deposited, individual colony forming units being deposited, CFUs. And from there, we grow them up. Now, we can say, hey, all of the cells of this particular colony are of one species. We can aseptically take our loop, scoop up that one colony that is now a pure culture, and introduce it into fresh media. That way, we know that everything that is in that new freshly inoculated media is from the same colony. They are of the same species. They are a pure culture from a mixed culture. All right, so microbes are really cool to grow up in a lab, but out in the wild, out in nature, where they would normally be found, they do not, absolutely do not grow in pure cultures. There are numerous different species living together, and they're going to be influencing one another because they are competing for the same nutrients. Sometimes they're competing for physical space as well. So we're going to begin talking about the growth cycle of bacteria. Before we move into the actual phases of the cycle itself, I'd like to go back and refresh our memory on what it means to divide when you're a prokaryotic cell. So all of our prokaryotic cells are single-celled organisms. They are going to reproduce through binary fission. We are going to have our single chromosome in our nucleoid copied. Do you remember which enzyme is going to copy that chromosome? What's its name? The name of the enzyme that replicates the DNA is called DNA polymerase. So within our bacterial cell, DNA polymerase copies the chromosome, and we have 2 copies within that cell. And then we undergo binary fission and we physically divide the 2. Each of those 2 then copies their chromosome, divides. We do this time and time and time again. Now, it's important to note that eukaryotic cells undergo a type of cell division as well, but it's slightly different because eukaryotic cells are larger, more complex, and they have a nucleus. So the name of the type of cell division that eukaryotic cells are going to do is called mitosis. So just a heads up in the terminology. For our course, we don't cover the detailed steps of mitosis, but if you have had a biology class either in college or back in high school, it is very likely that you learn the steps of mitosis. With prokaryotic cells, it is much faster and more simplistic. That's one of the reasons that they can undergo binary fission so quickly. All right, so let's talk first about like the maximum potential that our prokaryotic cells can do in terms of dividing. So when nutrients are ample, we have what is called exponential growth in our bacterial cells. This is where our population of cells is going to double at a pretty fixed rate, and it's a different rate for different species of bacteria. The exponential growth for bacteria is much quicker because they can divide about every 20 minutes than the rate of something, say, like Mycobacterium species. Recall that Mycobacterium have that mycolic acid, that waxy component to their envelope. So they have a slower growth rate because they have a harder time getting nutrients in. Okay, so there's a scale at what the rate is, but exponential growth basically means that we are having binary fission taking place as quickly as it possibly can. And when we plot exponential growth just in sheer numbers of cells, we get what is called a J-shaped curve. So that's what we have depicted over here. So we have time down here, and they're showing you in days. And then over here we have actual numbers, so the number of cells per milliliter. An exponential growth curve starts off with pretty slow growth. 1 becomes 2, 2 becomes 4, 4 becomes 8. But when we get to those bigger numbers and we're doubling, and then we're doubling even bigger numbers, we get really fast growth once we pick up that pace. So when we start talking about this doubling time, that's called the generation time. It's this division at a constant pace. We are only going to see this when our nutrients are in ample supply. Think of them as basically being unlimited. So the bacteria are dividing at their maximum speed. Now, when we start looking at environmental conditions, the media that they're growing in, whether or not the temperature is ideal, whether or not the pH is ideal, those things all do play into that, and we'll talk about that in part B. But the idea here is that in the real world, when you have other microbes that are competing for your nutrients, we cannot see this indefinite exponential growth. You can't just continue dividing and dividing. If not, the whole world would be covered with bacteria. So we start seeing that limits to those nutrients is important. We are not only going to be running out of nutrients in a given area, as those nutrients are being processed, waste byproducts that are usually, they may be either toxic or they may just not be good. They may not be ideal. But those waste products are harmful to the cells, and so we're not going to see that exponential growth forever and ever. When we want to look at this pattern of how quickly we get to our exponential growth and then at what point do we start running out of nutrients, we can perform what is called a batch culture in a lab. Think of a batch culture as a closed system, okay? So essentially what it is it's taking nice, fresh media that has ample nutrients, those essential nutrients, those growth factors, whatever the species needs, and we're going to take that given amount of media and we're going to inoculate it. We are purposefully going to introduce a given species. When we do a batch culture, we're going to use a pure culture to inoculate our media because we're interested in that particular species' growth patterns and how quickly they're going to utilize those nutrients and make harmful waste products within that environment. When we're doing a batch culture, we never refresh the media. So if we're trying to keep a pure culture for a long time, we have to provide new media to that pure culture. Well, in a batch culture, we don't. We basically say, this is what you get. Let's see how you make use of this amount. So what happens is that over time, as the nutrients begin to utilize, as the cells begin to utilize those nutrients, the media is going to begin to deteriorate. We're going to have fewer nutrients that are available. Those waste products build up, and we see changes in the cells within that batch culture. They are going to follow a predictable pattern called a bacterial growth curve. So each species has a given length of time that they will run through this curve pattern. Sometimes it's over a matter of days. Sometimes it can be weeks. Again, it's really going to be dependent on that particular species, but they're all going to have 4 sections to their growth curve. They're going to have a lag phase, they're going to have a log phase, they're going to have a stationary phase, and a death phase, always in that same order. And the reason that we see these changes in the number of living cells is because of what's going on within this media. Okay, so let's start, and real quickly, just because we emphasize the J-shaped growth curve, that particular graph showed just the numbers of cells. This one has changed the scale, so they're using a logarithmic scale. Again, the conversion of the two is not something that I'm interested in you knowing, but understand that when you plot a J-shaped exponential curve on a log scale, it becomes straight. Okay, so that's what this linear growth represents right here. So this straight increase is because we have a log scale rather than seeing that slow curve. Okay, so let's begin by looking at this darkest blue area down here, our lag phase. So this is when we've taken our batch culture and we've just introduced those first few cells. During the lag phase, our cells are living, but they're just not actively dividing. They're not undergoing binary fission. What they're doing in the lag phase is detecting the different types of nutrients that are available within their environment. So bacteria are super cool in the fact that they can metabolize different types of nutrients. In order to metabolize and use those nutrients, they need to turn on the correct set of genes that are going to cause the enzymes to be made to metabolize those nutrients. They will want to turn off the genes that code for enzymes for nutrients that aren't available. The idea here is that bacteria are really efficient. They're going to make the enzymes that they need to metabolize only the nutrients that are there. They're not going to waste energy making enzymes to metabolize nutrients if those nutrients aren't in their environment. So during this lag phase, we shut down the expression of the genes for stuff we don't need, and we express the genes for the enzymes that allow us to metabolize the nutrients that are actually there. So they're just figuring out what's going on at this point. They're getting ready to divide, but they're not there yet. So once they figure out the nutrients that are there, they've got those enzymes made, they begin to divide and divide and divide. Because we still have fresh media, we're going to see nearly exponential growth. And this is where we see that straight logarithmic line reflecting that exponential growth. Here, we are functioning at maximum capacity. All of those cell components, those macromolecules, are being made at a constant rate because nutrients aren't a limiting factor, but they will eventually become one. So when we hit the stationary phase at stage 3, what do you notice about our number of microbes? Well, it's flattened out. So during our stationary phase, we see a flat shape to our curve because there isn't a change in the overall number of living cells. What happens is that we begin to deplete our nutrients and those toxic waste products begin to build up. So at this point, we are having just about as many cells made through binary fission as we do number of cells that are dying off due to lack of nutrients or those waste products. So there's no net gain in the number of cells. At this point, we may see the cells undergo some physiological changes if they have the capacity. So if we have an endospore producing species within either the genera Bacillus or Clostridium, this is the time that they will begin to make those dormant, hardy endospores. We may see overall decrease in cell size when we're undergoing binary fission. We simply don't have as much nutrients and as many things available to have like a nice big cell. So sometimes they're smaller in size. We may have stress-resistant enzymes that are being made. They may also just make minor adjustments to allow them to survive in this, what's considered to be a more harsh environment. When we get to the 4th stage, if we don't deplete the media, we will eventually see all the cells die. So when we begin to see more cells dying than are being produced through binary fission, this is when we're in the death phase and we see that slope downward. At this point, DNA polymerase, our enzyme that is supposed to be copying that chromosome for each of our daughter cells, it's running out of energy and it gets sloppy, and DNA polymerase doesn't do its job as well during the death phase. And so it's not uncommon for mutations or changes in the DNA to occur because DNA polymerase isn't doing its job. So there is a phenomenal textbook out there that has a great animation. However, we don't use it. So the animation that I would like to show you is actually one that is used by a different professor as part of their online lecture series. But it's great at showing the number of cells and where we're at in this growth curve. We're not going to watch the whole thing. The professor who is narrating this goes on to talk about the relationship between our exponential curve and that logarithmic scale. We don't need to know that.
Bacterial cells do not divide by meiosis and mitosis. Instead, most bacteria grow asexually by a process known as binary fission. In this process, the bacterium copies its chromosome, elongates, and splits off into 2 daughter bacteria with a genetic makeup identical to the parental bacterium. Each daughter cell can continue the doubling process. This process happens rapidly, so the number of bacteria present in a solution can increase very quickly. When dormant bacteria are transferred to a fresh medium with plenty of nutrients, they spend some time switching their metabolic machinery from the dormant state to an actively growing state. The lag phase can last from less than an hour to days, depending on the species of bacteria. During the log phase, or exponential growth phase, the bacteria are actively undergoing binary fission. During the log phase, the bacteria double their numbers every generation period. The generation period can range from 20 minutes to days, depending on the species. As long as there are plenty of nutrients and little waste buildup, the bacteria will continue growing exponentially. It is convenient to represent the growth of the bacteria on a logarithmic graph, because the number of bacteria increase very rapidly with each subsequent generation. When the log of the number of cells is plotted versus the generation, the graph is linear. After the bacteria have grown for a while, and they are starting to deplete the nutrients in the growth medium, some of the cells begin to die. The stationary phase is reached when the number of new cells equals the number of cells that are dying. After the bacteria have depleted most of their nutrients and their waste buildup is getting too great, the bacteria begin to die in larger numbers than are made. If the death phase is carried on for a long time, most of the cells will die. If a cell is to survive, it must switch its metabolism back to a dormant state because there are not enough nutrients available for any more growth.
This is where we're going to stop for part A. We'll pick up part B on the next slide, slide 13.